Students, welcome to Neet Wizard. So here we are going to discussing about the sexual reproduction in flowering plants, and here the audio notes for this chapter. In this video we will discussing about only that very important points and extractive question coming points from the NCERT. So let's start it from the pre-fertilization events. Several hormonal and structural changes result in the development of a flower. Unflowering bear the flower buds and then the flowers. Flowers are the reproductive parts of a plant. In the flowers, the androecium male reproductive part and the gynoecium female reproductive part develop. Androecium. The androecium consists of whorls of stamen. The stamen consists of the filament, long and slender stalk and anther, which one is bilobed structure. Filament is attached to the thalamus or the petal. Anther. A typical anther is bilobed and each lobe is dithecous, consists of two theca. Theca are separated by a longitudinal groove running lengthwise. The microsporangia are located at the corners, two in each theca. They further develop to form pollen sacs which contains the pollen grains. Now, structure of microsporangium. The microsporangium is surrounded by four wall layers: epidermis, endothecium, middle layer and tapetum. Epidermis is the outer layer and the middle layer, and the tapetum is the last and the inner layer. The outer three layers are protective and help in the dehiscence of anther to release the pollen grains. The tapetum provides nourishment to the developing pollen grains. In the young anther, the sporogenous tissue forms the center of each microsporangium. Now, microsporogenesis. It is the process of formation of microspore from PMC pollen mother cells. As development occurs in the anther, the sporogenous tissue undergoes meiosis to form microspore tetrad. Each cell of sporogenous tissue has capacity to give rise to a tetrad. Hence, each cell is a potential pollen or PMC. Now, as the anther matures, the microspores get detached from each other and develop into pollen grains. Now, pollen grains represents the male gametes and are spherical, having a two-layered wall. Outer layer is axine, hard layer made of a sporopollenin, which is extremely resistant and can withstand high temperature, acidic and alkaline conditions and enzymes. Inner layer is entine, which is thin and continuous layer made up of cellulose and pectin. Mature pollen grain contains two cells: vegetative cells and generative cells. Vegetative cells is large with irregular nucleus, contains food reserves, and the generative cell is a small and floats in the cytoplasm of the vegetative cell. In 60% of the angiosperms, pollen grains are shed at two-celled stage, while in other generative cell undergoes mitosis to form two male gametes three-celled stage. The viability of pollen grains after they are shed depends upon temperature and humidity. It ranges from 30 minutes to few months. Gynoecium and formation of female gametophyte. The gynoecium represents the female reproductive part of a flower. It may be monocarpellary, one pistil, or multicarpellary, many pistils. In multicarpellary, the pistils may be fused in one is known as a syncarpus, and the pistils is free, so known as apocarpus. Each pistil consists of a stigma, style, and the ovary. Stigma receives the pollen grains, style, elongated slender part below the stigma, a very bulge vessel part containing the placenta, which is located inside the ovarian locule and the cavity. The placenta contains the megasporangia or ovules. Megasporangium, the ovule is attached to the placenta by the funicle. The junction of the ovule and the funicle is called hilum. Each ovule has one or two protective layers called integuments which cover the rest of the ovule except for a small opening called micropyle. The chalza lying on the opposite side of the micropyle and represent the basal part of the ovule. Nucellus is present within the integuments and contains reserved food. The embryo sac or female gametophyte is located within the nucellus. Now megasporogenesis. The megaspore mother cell gets converted into megaspores by the process of megasporogenesis. The megaspore mother cell is large and contains a dense cytoplasm and a prominent nucleus. It undergoes meiosis to produce 4 megaspores. Female gametophyte. In most flowering plants, only one megaspore is functional while the other 3 degenerate. The single functional megaspore develops into the female gametophyte. This kind of development is called monosporic development. The nucleus of the functional megaspore divides mitotically to form 2 nuclei which move towards the opposite ends, forming 2 nucleate embryo sac. 2 more mitotic division ensure leading to the formation of 4 nucleate and 8 nucleate embryo sacs. After the 8 nucleate stage, the cell walls are laid down and the typical female gametophyte embryo sac gets organized. 6 of the 8 nuclei get surrounded by the cell wall and the remaining 2 called polar nuclei are situated below the egg apparatus in the large central cell. 3 of the 6 cells are placed at the micropylar end and constitute the egg apparatus. 2 synergids, 1 egg cell. The synergids have special thickenings of the micropylar end. These are together called the filiform apparatus. It helps in the leading the pollen tubes into the synergid. 3 cells are at the chalzal end and are called antipodal cells. A typical angiosperm female gametophyte is 7-celled and 8 nucleated of maturity. Now, pollination. It is the process of transfer of pollen grain from the anther to the stigma. Depending on the source of pollen, pollination can be divided as follows. Autogamy. It is the transfer of pollen grains from the anther to the stigma of the same flower. Autogamy requires the anther and the stigma to lie close. It also requires synchrony in the pollen release and stigma receptivity. Plants like viola, oxalis and the camelina produce 2 kinds of flowers, chasmogamous flowers with exposed anther and stigma and clistogamous flowers which do not open at all and only autogamy occurs. Gynetogamy. It is the transfer of pollen from the anther of one flower to the stigma of another flower. In the some plant genetically it is similar to autogamy but it requires pollinating agents. Xenogamy. It is transfer of pollen grains from the anther to the stigma of a different plant. Pollination causes genetically different type of pollens to be brought to a plant. Now, agents of pollination. Plants use air, water, abiotic agents and animals biotic agents for pollination. Pollination by wind. It is the most common form of abiotic pollination. Plants possess well exposed stamens and large feathery stigma. Pollen should be light and non-sticky to be carried easily by winds. Wind-pollinated flowers often have single ovule in the ovary and numerous flowers packed in an inflorescence. It is common in grass. Pollination by water. It is rare in flowering plants except for some aquatic plants like Vallisneria and Hydrilla. In most water pollinated plants, the pollen grains are long and ribbon like and are protected from wetting by mucilaginous coverings. In a majority of water plants like water hyacinth and water lily, flowers emerge above the water level and are pollinated by insects. Pollination by animals. Majority of flowering plants use butterflies, bees, wasps, etc. for pollination. Most of the insect pollinated flowers are large, colorful, fragrant, and contain nectar to attract the animal pollinators. These are called floral rewards. Floral reward can be in the form of providing safe places to lay eggs, as an example the tallest flower Amorphophallus. A symbiotic relationship exists between the plant yucca and its pollinator moth. The moth is dependent on the plant since the moth deposits its egg in the locule of the ovary of the plant, and in return the plant is pollinated by the moth. The pollen grains are sticky and get stuck to the body of the pollinator. Outbreeding devices. Repeated self-pollination leads to inbreeding depression. Plants have developed methods to prevent self-pollination. Autogamy is prevented by following ways like pollen release and stigma receptivity not coordinated. Different positioning the anther and the stigma. Production of unisexual flowers. Ways to prevent both autogamy and gytonogamy. First is presence of male and female flower on different plants such that each plant is either male or female as a dioecious. This mechanism is present in several species of papaya. Pollen pistil interactions. Pollination does not always ensure the transfer of compatible pollens. Hence, the pistil has the ability to recognize the right type of pollen to promote post-pollination events. If the pollen is of the wrong type, the pistil prevents pollen germination. This interaction is mediated by chemical components of the pollen and the pistil. Pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The pollen tube reaches the ovary and enters the ovule through the microfile. Then through the filiform apparatus it reaches synergids. In this way, the pollen tube grows. Artificial hybridization and double fertilization. Artificial hybridization. It is a method to improve crop yield. In this method, it is essential to ensure that the right kind of the pollen grains are used and the stigma is protected from unwanted pollen grains. It is achieved by emasculation. The anther is removed from the bud if the female parent bears bisexual flowers. Second is bagging. The emasculated flower is covered by a bag so as not to allow contamination of the stigma by unwanted pollen grains. When the stigma of the bagged flower become receptive, the collected pollen grains are dusted onto the stigma and then the flower is rebagged. If the female parent is unisexual, emasculation is not necessary. In this case, the female bird is directly bagged, and when the stigma turns receptive, suitable pollen grains are dusted onto it so as to follow germination. Double fertilization. When the pollen grains fall on the stigma, the pollen tube enters one of the synergids and releases two male gametes. One of the male gametes moves towards the egg cell and fuses with it to complete the syngamy to form the zygote. The other male gamete fuses with the two polar nuclei and the forms triploid primary endosperm nucleus. This is termed as triple fusion. Since two kinds of fusion, syngamy and triple fusion, takes place, the process is known as double fertilization and it is characteristics of the flowering plants. After triple fusion, the central cell becomes the primary endosperm cell. The primary endosperm nucleus gives rise to the endosperms, while the zygote develops into embryo. Now, post-fertilization events. It includes development of endosperms and the embryo and maturation of ovules into seeds and ovaries into fruits. Formation of endosperms. The endosperm develops before the embryo because the cells of the endosperm provides nutrition to the developing embryo. The primary endosperm nucleus repeatedly divides to give rise to free nuclei. This stage of development is called free nuclear endosperm. Cell wall formation occurs next, resulting in a cellular endosperm. The endosperm may be either fully consumed by the growing embryo as in a pea and beans or retained in the mature seed as in coconut and castor. Development of embryo. The embryo develops at the microfilar end of the embryo sac where the zygote is situated. The zygote gives rise first to the proembryo and then to the globular heart-shaped mature embryo. A typical zygote embryo consists of an embryonal axis and two cotyledons. The portion of the embryonal axis above the level of cotyledons is called epicotyle. It contains the plumule shoot tip. The portion below the axis is called hypocotyle. It contains the radical root tip. The root tip is covered by the root cape. In a monocot embryo, there is only one cotyledon. In grass, it is known as scutellum and is situated at one side of the embryonal axis. At its lower end, the embryonal axis has the radicle and the root cape enclosed in the coryza. The epicotyle lies above the level of the scutellum and has the shoot apex and leaf primordia enclosed in hollow structures called saloptiles. Seeds and fruits. Development of seeds. It is the last stage of sexual reproduction in angiosperms. Seeds are the fertilized ovule that are developed inside a fruit. A seed consists of seed coat, cotyledons, embryonal axis. Seeds may be albuminous endosperm present as in wheat and maize or non-albuminous endosperm absent since it is consumed by the growing embryo as in pea and beans. Some seeds such as black pepper and wheat have remnants of nucellus known as perisperm. The integuments of ovules harden to form the seed coat and the microfile facilitates the entry of oxygen and water into the seed. As it loses moisture, the seed may enter dormancy or in favorable conditions exits it germinates. Development of fruit. The ovary of a flower develops into a fruit. The walls of the ovary transforms into the walls of the fruit pericarp. Fruits may be fleshy as in mango and orange or can be dry as in groundnut and mustard. In some plants, floral parts other than the ovary take part in fruit formation as in apple and strawberry. In these, the thalamus contributes to fruit formation. Such fruits are called false fruits that develop from the ovary are called true fruits. Some fruits develop without fertilization and are known as parthenocarpic fruits, for example, banana. Now, apomixis and polyembryony. Some plants produce seeds without fertilization. This process of seed formation is known as apomixis. Apomixis is a form of asexual reproduction mimicking sexual reproduction. In some species, apomixis occurs as the diploid egg cell is formed without meiosis and develops into embryo without fertilization. In some varieties of citrus and mango, the nucellus cells divide and protrude into the embryo sac to develop into embryos. In such cases, each ovule may contain several embryos, and this condition is called polyembryony. Apomixis is important for producing hybrid varieties of fruits and vegetables, and also for increasing crop yield multi-fold. Now, this chapter reproduction in flowering plants is complete now.